Hi, how are you doing? I hope you are taking good care of yourself, and I hope you are taking good care of your studies also. Um, we are to adjust according to the time. India is facing a bad time, so we are to adjust accordingly. Good time will come. Rest assured, we shall win over this situation. We start today with a new topic, and the new topic is energy disturbances caused by different drugs and different chemicals in antibiotics. Energy differences. When we talk of energy, immediately our attention goes to E is equal to mc square. Our attention goes to the three basic laws of thermodynamics. Energy cannot be destroyed, cannot be created. Energy goes from higher level to lower level. Energy leads to heat, enthalpy. There is something like free Gibbs energy. There is something like H. There is something like enthalpy. So, with this basic background of physical chemistry on energy, when we go to another aspect of energy, that from where energy comes, it comes from sun. It is fixed by the autotrophs. From autotrophs, it goes to heterotrophs, saprotrophs, and so on. And again, in this way, a energy cycle, which we call as biogeochemical cycle or energy cycle, that is maintained. The same thing happens in the body also. As we have got a energy budget for a forest, or we have got a energy budget for a river or for ocean, for a country, for a town, similarly there is an energy budget in your body also. All functions, all tissues, all organs are working on a particular energy budget, and that energy budget is maintained by a energy balance, and that is called as energy homeostasis. And similarly, each cell of your body is having its energy budget. There are few cells which are latent most of the time. There are few cells or systems which are very active. You sleep, but your heart does not sleep. The heart muscles keep on working, right? Your legs and arms are sleeping, they are not working. Skeletal muscles requirement for oxygen are negligible, but the oxygen requirements for the heart are equally good as they had been when you were up, you were wake up. So in this way, what do you find? Oh yes, this how this energy in the cell is maintained, and that energy in the cell is maintained by an organelle which we call as mitochondria. You must have been reading mitochondria in BSc also, in intermediate also. We say mitochondria is the powerhouse of the cell. Ah, yes, it is the powerhouse of the cell. The same concept we shall proceed further. How that mitochondria works as a powerhouse of the cell, right? The powerhouse means it generates energy. From where it generates energy and in what forms of it generates energy? The energy is generated in the form of a protein or in the form of an enzyme ATP. And from where this ATP molecule comes? This ATP molecule comes from ADP and the phosphate molecule. So ADP plus P, they join together with the help of an enzyme ATP synthetase, and the synthesis of ATP takes place. In the same way ATP used, again it is broken down to ADPP, again it is with the help of the ATP synthase it is synthesized, and this cycle continues in the cell. It's a regular cycle going on in the cell, in a normal cell, behaving this ATP synthetase. Where does it reside? It resides in mitochondria. So all these things are happening in the mitochondria. It's the mitochondria which is generating the ATP molecules. So this mitochondria, how does it generate, and what are the different functions of mitochondria in a normal cell? Mitochondria is responsible for uptake of the substrate. The substrates come from glucose in the form of the pyruvate, in the form of fatty acids, and these pyruvate and fatty acids, they cannot be carried from. You see, mitochondria has got a structure. There is a outer mitochondrial membrane, there is a inner mitochondrial membrane, there is a matrix. Matrix is filled up of cristae, and there is a space between the outer mitochondrial membrane and the inner mitochondrial membrane. We call it as intermembrane space. The outer membrane allows many things to come in, but the inner mitochondrial membrane does not allow many things to enter into the mitochondria. It is a gated channel, and that is controlled by the electrical gradients which are present over there. Nothing can enter into the matrix of the mitochondria without crossing the inner mitochondrial membrane. And how it will cross the inner mitochondrial membrane? It cannot cross like this. So there are certain molecules which are transporting the molecules from intermembrane space to the matrix, and these are known as carnitines, and we call them as the carnitine as a carrier, and they are also referred to as acetyl carnitine. Acetyl carnitine, because they are helped by an enzyme, acetyl coenzyme A, and this acetyl coenzyme A is present in the matrix, and this acetyl coenzyme A is responsible, it follows a cycle which we call as citric acid cycle, and the citric acid cycle gives rise to a molecule, to a protein which we call as NADH. So this happens in the matrix. So there is something which is taken from the outer environment of the mitochondria, it enters into the mitochondria, it enters into the matrix, and the NADH molecule is being formed. And then what do we find? That there is again the NADH generates oxygen and water, changes into oxygen water, and there is a molecule which we call as ubiquinone, which is present at the membrane level, and these processes are referred as electron transport chain. You know that mitochondria plays a very important role in electron transport chain. So this is how the electron transport chain occurs, starting from acetyl coenzyme A and leading to the ubiquinone. And there is another very important function that occurs. We call it as the protonephric activity or protonation, or in very simple words we can say that it is the protein pump. So that protein pump works, protonation works in between the outer membrane, mitochondrial membrane, and the inner mitochondrial membrane. And this proton pump is responsible for maintaining the transport, the efflux and efflux of the molecules inside the cell and outside the cell. In addition to this normal functioning of the mitochondria, there are certain concepts which you should remember. There are a lot of work is being done on mitochondria. Every year, a conference, international conference, is organized in Berlin, Germany, every year, and it is called as Mitochondria Conference. And the people who work on mitochondria, they come there and they discuss, they deliberate upon on their research achievements. We are nowadays, when we talk of mitochondria in terms of toxicology, or when we talk of the disturbances caused by energy, in energy homeostasis by genobiotics, the one and the same thing, that how the mitochondria are being affected by drugs or chemicals. So when we do talk of this, we talk that yes, there is a concept of mitochondrial homeostasis, that how mitochondria is able to maintain its balance in a acidic environment, in a hyper alkaline environment, in an environment which is having something exogenous in it. So that is how mitochondria is able to do. Many times we perform some experiments and we find mitochondria changes morphology. Sometimes mitochondria is oval, sometimes it is like a spindle shape, sometimes it is egg shaped, sometimes it acquires another shape, elongated shape. Sometimes the cristae becomes enlarged, sometimes the cristae becomes shrunken, sometimes the outer membrane and the space between the outer membrane and inner membrane also changes when we compare it with a control observation. So these things are going on. Any very important issue is that if the mitochondria dies, what happens to it? It is called as mitophagy. Like autophagy, it is called as mitophagy. So mitophagy, mitochondrial homeostasis, mitochondria, role of mitochondrial cancer. Recently, I read an article, very interesting article on mitochondria and cancer. How the mitochondria plays a role in carcinogenesis. And it was a very interesting work done by Wallace. There is a person, Wallace, who published very recently, maybe just a few months back. He published that article, Wallace, and I read it just last week or maybe 10 days before. So a lot of work is being done on mitochondria. How the xenobiotics they can affect mitochondria, it has also been elucidated. It has also been studied in many laboratories for different xenobiotics. It may be inorganic, organic, it may be pesticide, it may be metal, it may be anything. So the people have studied that in their laboratories, and what do they conclude? They divide this entire affair into few headings. And the first heading is that the xenobiotics they inhibit fatty acid uptake and the beta oxidation. Fatty acid uptake and beta oxidation, that is disturbed by the xenobiotics. The second is inhibition of the citric acid cycle. The citric acid cycle is inhibited, and the third one is the protonoferric activity, that is uncoupling of the electrons, that is done by the proton pump. Then next is the accumulation of small xenobiotics in the matrix. Nanoparticles, for example, I was reading today morning, nanoparticles get deposited in the matrix of the mitochondria. Imagine the matrix, which is having the crystal like this structure, and a particle which is having the size of less than 100 nanometers is able to cross the inner mitochondrial membrane and get deposited into the matrix. So many of the xenobiotics they get deposited into we shall discuss each and every step with a suitable example. They get deposited in it. The next issue is the inhibition of the electron transport, that the xenobiotics they disturb the electron transport. I talked to you the normal function of the motor mitochondria is to perform the electron transport chain, and that electron transport mechanism is disturbed by many of these xenobiotics. Then many of these xenobiotics they are able to inhibit the synthesis of ATP by inhibiting the enzyme ATP synthetase. They inhibit the enzyme ATP synthetase activity, and the ATP is not formed from ADP plus P because there is no enzyme to catalyze that reaction. Then there is another mechanism which we call as the very important one in apoptosis and necrosis. When we talk of apoptosis, we shall be discussing it in length. This is the opening and closing of the mitochondrial permeability transition pore. So, where this MPTP exists, it exists in it is a hypothetical pore. It is known to exist in the inner mitochondrial membrane. And when in normal case, the inner mitochondrial membrane and MPTP in the membrane remains closed. It is that the gate is closed. But when these xenobiotics they interfere with, they see the story, they enter through the plasma membrane, they come to the cytosol, and from cytosol they are able somehow to manage the entry through the outer membrane, go to the intermembrane space, go to the inner mitochondrial membrane, and they somehow manage the we shall be discussing later, they somehow manage the opening of the mitochondrial permeability transition pore. And when this mitochondrial permeability transition pore MPT opens, there is the release of the cytochrome C from mitochondria into the protoplasm. The cytochrome C release is responsible for activating certain factors that lead to apoptosis or necrosis or cell death. And there is one more mechanism. This is very important, opening and closing of MPTP. Remember it, we shall talk it in details in apoptosis as well as in necrosis. Another important issue that is that how these xenobiotics can enter, can disturb the mitochondria. That is the binding of the xenobiotics with the molecule cardiolipine, and cardiolipine is present in the inner mitochondrial membrane, and these xenobiotics, like they enter into the matrix, they can bind with the cardiolipine molecule, and in this way they can another very important point is that these xenobiotics can deplete the oxygen availability to the mitochondria, and that we measure in many experimental conditions, in many while we observe the effects of many xenobiotics. That is called as a mechanism, we call it as oxidative phosphorylation, and it is called as oxphos, not fox, oxphos, OXPHOS, oxphos. You should remember it. What is oxphos? Oxphos is oxidative disturbances caused by xenobiotics through this mechanism. And when oxygen depletion occurs, you know what will happen? That will lead to no oxygen will be, or the less oxygen will be available to the heart muscles, the less oxygen will be available to the skeletal muscles, less oxygen will be available to the kidney, less oxygen will be available to the beta cells of the pancreas. So in this way, through mitochondria, many of these effects will be implemented, will be affected, will be manifested, I can say like this. So what happens is that the mitochondria remains a target of many xenobiotics. We call a target toxicity. We study receptors, that when a metabolite or toxicophore goes into the cell, it is received by the receptor. Similarly, everything cannot injure mitochondria. Everything cannot injure peroxisomes. Every xenobiotic cannot affect the lysosomes. So the mitochondria, you see, mitochondrial DNA is affected by xenobiotics. Lot of work is being done now on the effect of xenobiotics on mitochondrial DNA. You see, the mitochondrial membrane is peroxidized by the xenobiotics. We had discussed about the lipid peroxidation. It is not only at the plasma membrane level, it is at the mitochondrial membrane level also. We call it as mitochondrial lipid peroxidation. Mitochondria are responsible for generating the free radicals, reactive oxygen species. They come from mitochondria. And how do they come from? That they do come from the peroxidative mechanisms that occur at the mitochondrial membrane level. So in the same way as we were talking of the plasma membrane, we do talk of the mitochondrial membrane. We do talk of the damage to the mitochondrial DNA. We do talk of the the damage which is caused to the morphology of the mitochondria. The structure is affected, the function is affected. So these drugs and chemicals or their metabolites, once they are able to enter into the matrix of the mitochondria through protonophoric activity, through disturbing the proton pump in the mitochondria, they play a havoc with the cell, or they play havoc with the mitochondria. They disturb the energy production, they disturb everything, they disturb oxidative phosphorylation, they disturb oxygen supply, they disturb the enzymatic machinery. Therefore, we study, why do we study these effects on mitochondria? Because we want to study what is the role of mitochondria, or what will happen to mitochondria if the cell dies after an abuse by a chemical or a drug. So that is the basic point to understand why we are studying mitochondria, because we want to study the mechanism by which the cell dies, the cell, how the cell will die through the mitochondrial disturbances. That we will study in our next lecture. I think it will help you. Just watch this video again and again, and things will be clear to you. Best of luck. Do good.